next presentation is about the number and brightness method, which can be used to determine also the protein concentration. But instead of the motion or the diffusion coefficient, we measure the aggregation or oligomerization state of, for example, proteins, but could also be other things like lipids or so in cells. Okay, so if you want to measure the concentration of a certain molecule in a cell, you can do that provided you can fluorescently label it. For example, you express just the green fluorescent protein in a cell. And then you do, with the same settings on the same instrument, you do a reference measurement of a solution of EGFP, where you then know the concentration. So you know, for example, in this hypothetical scenario, a 10 nanomolar EGFP solution will give you 30 kilocounts per second. So if you then go to the cell and you have two regions A and B, in one region you have 30 kilocounts per second, and in another one you have maybe 90 kilocounts per second, you would conclude that in region A you have 10 nanomolar, and region B you have 3 times, so 30 nanomolar of that fluorescently labeled molecule. And this is fine, but it doesn't give you the size distribution. It doesn't tell you in which form the molecules exist. Are they all monomers, or are they dimers, or are they assembled into even larger complexes? So to get the oligomerization states, there are other methods, and one popular approach is via first resonance energy transfer, FRET, which is a very nice and very sensitive method, where you have a protein labeled with a donor and an acceptor. In the case of homo-FRET, those could actually be the same fluorophore, but most popular is the hetero-FRET, where you have a donor which is of a shorter emission wavelength that then overlaps with the excitation spectrum of the redshifted acceptor molecule. And this is a very sensitive method. As soon as you have the two molecules very close together within a distance of a few nanometer, and also other conditions such as the orientation are fulfilled, you can get energy transfer between the two, which will result in a signal that you can detect. So for example, reduction of lifetime of the donor and increase of the fluorescence of the acceptor. So this is very sensitive to the detection of the formation of pairs, but it does not tell you for any higher oligomers. I mean, there could be 4 and you have more energy transfer, but that would be hard to quantify. Another alternative is via image correlation spectroscopy, ICS, but this will only work if you have spatially well separated aggregates and if they move slowly. If they move fast, it will be difficult to apply this method. So what is number and brightness? What is it trying to do? So the idea is to provide a pixel resolution map of the molecular number and aggregation or oligomerization state of molecules in cells. The method that is used is we look at the first and second moment of the fluorescence intensity distribution in each pixel. And what can you use as an input? You can use the same raster scanned images that we just used for the RIGS analysis, but you can also use this, for example, with camera images obtained by total internal reflection microscopy or single plane illumination microscopy under certain conditions, which we'll go into a bit later. So what you get is number and brightness pixel by pixel maps. And then you can use cursor analysis, where you select certain regions of the cell and ask what is the oligomerization state. Or you can, in the NNB plot, select, for example, the dimers and then highlight them in the cell. So how does this method work? So the question is, how do we distinguish many dim molecules from few bright molecules, which in the end could give you the same average signal? So this scenario is illustrated in the top row. So in the first case, we have few but bright molecules because they are aggregates. So what will happen if you look at your confocal observation volumes, if the molecules are able to move, if a molecule is very bright, moving in and out of the observation volume, this will give you a large fluctuation of your signal as a function of time. In the other scenario, if you have very dim molecules, but a lot of them, if one of those molecules moves in and out of the observation volume, that will not change your signal very much. So the fluctuation will be kind of small. And this you can quantify using looking at the variance or the second moment of the fluorescent signal in each pixel. So yeah, if you have aggregates that have many fluorophores that are very bright, they will leave and enter the observation volume simultaneously, giving you this large variance, whereas the dim monomers will give you a smaller variance. Also, you can get the average number of molecules from this, similar to single point FCS or to RIGS, where the variance divided by the average intensity squared is proportional to 1 over the average number of molecules inside your observation volume. So this analysis provides a map of the average number of molecules and brightness for every pixel in the image. And the brightness unit is related to the pixel dwell time of your system. So typically you quantify it in the unit counts per dwell time per molecule. But later, if you have a standard, a monomeric standard, you can then just normalize by that and then say, okay, these are the monomers, these are the dimers, these are the trimers, and so on. Okay, so yeah, we are from the image series. Again, to apply this method, you need to collect a series of images in the same plane. Again, can be on the order of 100 images or so. And for each pixel, you calculate the average intensity, which is the formula on the top left. And you calculate the variance, which is the formula in the top right. And the brightness in each pixel will be average intensity by average number of molecules, which makes sense. So you can substitute that by the equation on the previous slide and get the brightness is the variance divided by the average intensity in each pixel. Because the average number is the average intensity squared divided by the variance. Okay. Now we have to take one important consideration, which is for the number brightness method, we rely on the molecules moving in and out the observation volume. If they don't move, we are not able to detect them. This is one advantage, for example, of the FRET. In the FRET, it wouldn't matter if they can move or not. You would still be able to tell if they're together or not. So if they move, we still have to be careful because the acquisition rate needs to be faster than the rate of the movement of the molecules. Otherwise, you will average out the movement and again, will not see any fluctuations. If you acquire your data on a laser scanning microscope, most of the time this condition will already be fulfilled and you don't have to worry about it. And the reason is that we move from one pixel to the next with our laser beam with dwell times on the microsecond time scale, which we just discussed for the RIGS. And in a cell, typically molecules don't move that fast. So the condition is well fulfilled. However, it can become a problem with camera-based systems, because now with cameras, you acquire the full image in one shot, but then you expose all the pixel for a longer time, maybe 10 milliseconds or even 100 milliseconds. So in this time, the molecules had plenty of time, for example, if they are moving fast in the cytosol, to move around and then average out your signal fluctuations. So in the camera-based systems, to overcome this problem, what you can do is, for example, you can use stroboscopic illumination. So you just flash the sample very fast, excite only for a period of, let's say, 100 microseconds or so, which is faster than the molecules can move between pixels, to not get this kind of motion blur. Okay. What contributes actually to the variance that we are quantifying? Well, there are two major contributions. One is the variance due to particle number fluctuations, and this is what we want to measure, what we want to get in the end. And this is equal to the brightness of the particle epsilon squared times the number of particles that we have. But then there are other variations, and the simplest one is the variance due to the detector shot noise. We're detecting single photons, so there's always going to be some shot noise. And so the variance of that is equal to the brightness epsilon times the number. So it scales differently in terms of brightness. So we can put the two together. So the combined variance is the variance introduced by the number fluctuations of the molecules and the variance introduced by the detector noise. But because they have different dependence on the molecular brightness, we can separate the two out. And then we can get n and epsilon, which is the true average number of molecules and the true molecular brightness. So to do the relation, we start with the brightness that we defined as the variance divided by the average intensity. Then we can split the variance into the two terms, the number fluctuations and the detector noise. Then we can substitute different dependencies on the brightnesses you were seeing on the last slide, and we see that we end up with epsilon plus 1. So meaning that if you have a system where you have molecules that are immobile, so they don't give you any number fluctuations because they cannot move, the brightness that you measure should be 1. And then once the molecules be moved, the effective brightness you measure should be larger than 1. This is also a good system you can do on your microscope to see if there are additional noise sources with your detector that you have to take into consideration. And then, so you can calculate the so-called true number of molecules and true molecular brightness, which is only coming from the molecules that move using those two formulas shown in the center here. So if there are regions of immobile particles, the number cannot be calculated because for the mobile fraction, the variance is equal to the average intensity because it's just a shot noise where this condition is fulfilled. Okay, yeah. So here's an example of the validation of the quadratic dependence of the variance of particle brightness. So this is a 20 nanomolar EGFP solution. And to increase the brightness, what was done is just the laser power was increased, which you can do very precisely in a very controlled way. And then the variance was calculated, and you see it fits this parabolic shape, fitting the dependence to the square of the brightness that you would expect. So how can we now identify and separate mobile and immobile fractions in a cell? So if the fraction is immobile, the variance divided by the intensity here should be locked in at 1 and should not change as you increase the laser power also. If you have the mobile fraction, of course, the the higher oligomerization state you have, the higher the variance will be. Or if you excite with more laser power, you will also get a higher variance. So this is illustrated on the graph on the right, where you have the brightness as a function of the measured counts per second on the photomultiplier tube. And the black circles are measurements on a slide with immobile molecules. And as expected, we get a brightness value of 1, which is independent of the number of counts per second, which was changed by changing the laser power. If we do the same measurement with a solution of EGFP, you see the brightness was larger than 1 because now we have this contribution from the number of fluctuations of the molecules. You also see that at very high count rates, it goes down, which is an artifact because you have to make sure that you're not saturating your detector. Otherwise, those considerations for the calculations will not be valid anymore. Okay, here's an example of fluorescent beads in a sea of 100 nanomolar of fluorescein, where the beads represent your immobile fraction and in the fluorescein are the molecules that move. So you see the intensity map on the top left, the brightness map on the top right. And now in the histogram, which is the brightness as a function of intensity, different populations were selected. The first are the ones with average lower intensity but higher brightnesses. And when you select this population, you see that the fluorescein is highlighted in the corresponding image by the red pixels. And if you select the portion which has higher intensity but lower brightness, this will correspond to the fluorescent beads in this image, which are immobile, but on the other hand, much brighter than the fluorescein. Okay, here is a monomer to octamer series. So different oligomers in solution were measured ranging from a monomer, and the next one was a dimer, then tetramer, and then octamer. And you can see that the brightness or variance over intensity nicely scales with increasing oligomerization state. And these were measured with a photon counting detector. Okay. A few more validations of the number and brightness principle. EGFP, the green fluorescent protein, again in solution. This is the 2D histogram of brightness versus intensity. You see that the average is slightly above 1. We can increase the measured brightness by changing the laser intensity, and it changes as expected. We can change the concentration and then also get a different average number of molecules, so that scales nicely as well. On the other hand, if we increase the concentration, the brightness does not change, which is expected because we still have a solution of monomers in the end. This also highlights an important point, which is that when you do your cell experiments, you should make sure that you keep the instrument parameters constant. Because if you're changing around the laser intensity, you will get a different brightness just because of that. And then you cannot compare your results anymore. So you need to make sure that all the measurements and the calibration measurements are all taken with the same intensity, laser intensity, same PMT gain, and so on and so forth. Okay, you already have seen that it works nicely with a true photon counting system. But what if you have an analog system? There are a few more additional considerations with an analog detection system that you have to take into account. And one is that you don't record photon counts in the end, but digital levels. So there is a conversion factor. Maybe one photon detected will give you 3 counts in your image. So there's this conversion factor from photon counts to digital levels. Then in analog systems, you typically have an offset. So even if you have no signal, nothing on your microscope, you will get some counts, which is the offset. And then the third parameter you have to consider is that you have additional detector variance, which is due to the noise of the analog system. Yeah, so to summarize, you have those three parameters: analog offset, conversion factor, digital levels to photons, and then the additional variance of the analog detector. And you can plug in those parameters into your equation, and you can then measure them, the reference measurement, quantify them, and then use them for the analysis. So if you don't change the PMT gain or any settings, then the S and sigma parameters should not change once you have determined them. And then you can calculate the number and brightness as before. So just to illustrate, this is detector characterization. So here's the typical response behavior of an analog detector with no sample present, just the dark current. On the y-axis, you have the number of counts in this histogram, and on the the occurrence of the histogram. On the x-axis, you have the digital levels, and then you see exactly those three parameters. You have an offset highlighted by this dotted vertical line. Then we have this kind of bell-shaped curve, which is the amplifier noise, in this case Gaussian noise. And then we have this exponential distribution of the signal levels, which is this digital level to photon, actual photon counts conversion factor. Yeah, so what you do on your instrument is you record the dark current, and then you plug it into the program, and then you can have the program determine those three factors. Just to show you as a comparison, for true photon counting detector where you don't need those correction factors, and same histogram, and you can see that basically all of your counts, almost all of them, the occurrence is 0. And then keep in mind the y-axis is a logarithmic scale. So here you have for 1 million occurrences of 0 counts, and then only 1 or 200 of 1 count, and even less for 2, 3, 4 or more counts. So if you have a true photon counting system, you are in luck because you don't have to worry about these correction factors, but it's always important to validate. Okay. Now to the application in cells. Here's a cell that's just expressing the green fluorescent protein. On the left top, you have the average intensity. On the top right or top center, you have an image or a map of the number of molecules. You can see that further inside the cell, you have more GFP molecules on average, and then down in the bottom center, we have the map of molecular brightness. And as you can see, it's the same everywhere, because in this case, the cell is just EGFP, it's just expressing EGFP, which is mostly monomeric if expressed at levels that are only in the nanomolar range. Okay, so let's go to an example. And this is penicillin in live cells, similar to the Riggs experiment. And in this case, the formation, assembly, and disassembly of the focal adhesions was investigated using the number and brightness method. So is the intensity in a given pixel of an image the result of many dim molecules, monomers, or is it a few brighter oligomers or aggregates? So here is the analysis of a region of a cell at the plasma membrane. On the left-hand side, you see the average intensity and color-coded from low to high counts, and you can identify the focal adhesions as areas where the measured intensity was very high. And then on the right-hand side, you see the brightness map, which indicates the oligomerization state. So the higher oligomerization, the hotter the color code in this case will be. So if you compare the two images, you see that, yes, you do have more higher aggregation states near the focal adhesions, but they don't exactly co-localize, which is better illustrated here in the following slide, where we have the number and brightness plotted to the histogram and then can select different aggregation states. So in this example, we selected the pixels that more or less correspond to EGFP in a monomeric form, and then color code these pixels in red. And you see, you highlight pixel mostly in the cytoplasm of the cell. Now we select pixels that show a much higher brightness. And you can see that those are near the focal adhesions, but not exactly overlapping with the focal adhesions. Here on the right-hand side, you can see the selection of the different distributions corresponding to monomers and then to higher aggregates with a cluster size, an average cluster size of about 8 molecules per complex. So why is this? Well, the reason is that once the adhesions are formed, the molecules, the penicillin molecules, don't move around anymore. They stay in this adhesion in the same spot until the adhesion is disassembled, for example, again. However, while they are incorporated in the adhesion, the oligomerization is increased and further increased. So at the edges of the focal adhesions, you have these higher order complexes forming, but also disassembling. So here you see a further illustration of this. 500 frames were acquired of this region and chopped up into bits of 100 frames, and each of those 100 frame averages, the number and brightness analysis was performed. And you see, for example, in the first chunk, we see those higher aggregates because the cell is disassembling this kind of adhesions. Why do we think it's disassembling it? Well, if you look at the average counts as a function of time, you see it goes down in this region. So that's why we concur that the cell was disassembling this adhesion. If you look at the cytoplasm just as a control, the intensity did not change, so it was not related to just photobleaching. If you look at other adhesions, you see that later during the acquisition of the image series, the intensity increased, and you can also see in the chunk of frame 400 to 500 that there are some small spots highlighted in red again that indicates formation of the penicillin clusters. So to summarize, number and brightness distinguishes between number of molecules and molecular brightness in the same pixel. The acquisition of number and brightness can be done on laser scanning microscopes, can be a commercial system, provided that you did the test to figure out if it's a true photon counting system or an analog system where you have to measure the correction factors. The other advantage is that you can actually analyze RIX data also with number and brightness. So it's kind of double purpose. You collect one data set and you can do both kinds of analysis. One thing notable is that RIX data you can typically analyze with number and brightness method, but number and brightness method you cannot necessarily analyze with RIX. And the reason is that for number and brightness, it actually doesn't matter what the pixel size is, which it does in RIX. So if you want to get the whole image within the whole cell, for example, within the field of view, you can with number and brightness. sacrificing the resolution, getting a larger pixel size, but then you can no longer analyze it with rigs. The mobile fraction can be separated in number and brightness because it has a brightness of equal to 1. And then you just saw the example where the NMB method was applied to those focal adhesions, looking at the packs in molecules and showing how larger aggregates form during the assembly and disassembly of those focal adhesions. Okay.